So this is a presentation about a course that I developed as a Mozilla Science Fellow starting 2015-2016, which is designed to bring open science and reproducible research skills to students in ecology, but hopefully to be spread beyond ecology eventually. So this is opening the field. So the idea is I want students to become better data science citizens while making their own work a lot more efficient. So I think that ecology is pretty important. We do a lot of work that determines how people interact with the environment. And we face a lot of big problems in ecology. So we've got climate change, we've got invasive species, we've got endangered species, we've got land management and conservation planning. And all of these problems end up having innate multidisciplinary and large-scale natures. We can't necessarily solve these problems if we're functioning in a single lab or in a silo, as I'm using the metaphor here. These are problems that we need to collaborate on. So if we're only focused on working alone, it doesn't necessarily get all the stuff that we need to do done. Ecology has a few challenges that we're facing, though. First of all, we are generating more information than we ever have in the history of the world. We are able to automate a lot of data collection. I mean, this is not even getting into genes and genomics. We are generating more data than we've ever known how to handle. We also have a problem in ecology in that a lot of how we do it has been determined by the technology that was available to us in the past. So for instance, how we publish our research hasn't changed appreciably since the advent of the printing press. We send our science off to a journal, and then the emphasis is on the paper rather than all the products going into developing the science. Now, of course, having the data, having the analysis is really essential if you want to reproduce science, and there's no reason that we can't include that now because we have the power of the internet and less limitations with storage. And then finally, we're dealing with the cowboy, what I like to call the cowboy myth. This is the idea that the best science, the most genius science is done by the lone cowboys out there communing with nature. But this ties right back to the silo problem. The problems that we face in ecology are not things that can be solved by a single person in a single lab. The problems that we have in ecology are big, so we have to work together. So what quantitative and computational skills do ecologists need in order to deal with these problems? I'd like to argue that, this is not all about programming. This is also about the soft skills that you need. So this is roughly a syllabus that you'd see for any quantitative ecology course that you'd have offered anywhere in the US. These are sort of the common elements. You'd want to see something to do with data management, data cleaning, data documentation, an introduction to R or some programming environment, manipulation of data in R, and data processing using basic programming skills, then plotting, introductions to modeling and multivariate methods, and fitting simple models, and some sensitivity analysis. And then a lot of courses go on to have sort of their own special flavor, but this is probably the common elements of every quantitative ecology course that you'd see in most of Western academia. And there's a lot of people who have been trying to get these pieces of information into people's minds. And so there's the biocenters, the NSF biocenters that have had a lot of training foci. There are courses that are focused on training biologists in stats and programming. Then there's short courses in independent entities like data carpentry and software carpentry. There's the study groups run by the Mozilla Science Lab, and of course, the LTER program is very keen on getting students and researchers involved in sustainable management of their data. But how do we make it stick? This is a picture from the course that I taught, and I'm going to tell you a little bit more about the methods that I used. This course was centered on a project-based learning approach. The idea here is, well, when you learn to swim, you don't learn to swim in a classroom, but you also don't learn by being dropped in the ocean. You use a controlled immersion in the local swimming pool, and you learn to swim that way. And so the idea here is you use real data, but in a very controlled environment, and essentially go through the whole motions of all the motions of developing a scientific workflow using this real data set in working in groups, very much like we do in labs, but in a very supported environment. The idea here is you adopt a growth mindset. When students see each other struggling with problems or working through issues, they get to see that it's not just, oh, I know how to do this, it's we're all learning. They get to directly apply the skills that they're learning about in class. Because it's a project using real data, they get increased investment in the project. So they can see that they're solving a real problem, and so they care about it. And then this is the key here. It becomes an outlet for underutilized data. So there's always cast-off data sets produced by all aspects of science. And these data can be used to support this course, and it helps actually move forward these file drawer data sets so they aren't lost to science. And so in addition to this project-based approach, we want these students to develop all of these hard skills. And so these are skills that I already outlined in the syllabus. But in addition to hard skills, we want them to see the why of all of why they're developing these hard skills. And I think that by and large, technology use is a means to develop soft skills. For example, when students have to use data sets that are produced by multiple users, they have to think very hard about how they're going to incorporate their philosophy on scientific authorship. And so in this class, students engaged in discussions, weekly discussions. So we talked about scientific authorship, we talked about software and science, we talked about copyright, licensing, reproducibility, publishing, and then some bigger modules on communicating science through visualization. So really how we engage with our data and how we bring the data to the public. And then citizen science and managing collaborative projects. So these are all technologically enabled soft skills, I would argue. And I think that these are the things that we really want students to be able to take out of the course. So using all of this reasoning, I developed a prototype for learning data science using some LTER data. So I have been studying the lady beetle communities at Kellogg Biological Station in southwestern Michigan since about 2012. But this is part of a long-term study that was established in 1989. We've been watching these communities and these lady beetle communities in 10 different plant habitats. Well-replicated design. But in 2004, the project had switched PIs, and bycatch, the technician at the time decided to start recording bycatch in our traps. And so he started recording, among other things, fireflies. And so from 2004 to 2015, we captured 17,000 fireflies. We actually have the longest systematic observation of fireflies known to science that was just in a bycatch data set associated with this LTR site. So I thought this presented quite an opportunity. So I told my students about this data set, and I had them brainstorm questions that they could ask of it. So questions like, where do we find fireflies? What plant communities are they associated with? And does it change over time? And so they asked all these questions. Where do we find fireflies? When do they peak? What factors affect firefly activity? And so on and so forth. And so in this presentation, I'm going to focus in on these two questions here. I'm just going to present our data and our workflow associated with these questions. But we actually did explore all of these different questions. over the course of the class. So this is a little bit of movie magic. This is probably the hardest part. We took the data and we cleaned it and we documented it and we poured it into RStudio. So this is the equivalent of bringing a fully baked cake out of the oven on a cooking show. It's a little bit more complicated than that, but this does take a fair bit of time. So if you're interested, you can go into the details in our course notes. We used RStudio as our primary development environment to do all of our work, which of course runs R. So why do we use R? Well, first of all, it is an open source software package that is freely available and maintained by the community, but also because it's rapidly becoming the standard statistical engine in ecology. So you can see that over time it's gained quite a bit of popularity, and in fact, as of about 2014, it looked like it is starting to overtake the number of references to SAS in citations from that year. It also produces very nice graphics, and because it's a command line based program, it allows for automation of our work and also reproducibility. So as you can imagine, when you're clicking through an interface, it's very hard to keep track of all of your click points, but when you have a script, it gives you a full recipe of exactly what you've done, making it easier for the next person to take it up and do it again. Another nice feature is it interfaces with GitHub. RStudio interfaces with GitHub, allowing us to keep version control of our code. So our complete class analysis is available on my personal GitHub, so you can click through and check out that link. So the first thing we did after bringing the data in and cleaning and documenting it is we explored the data. So the idea here is to find patterns, test for errors, and uncover the underappreciated biology within the data. So I wrote a blog post about this, and incidentally, it was my most popular blog post ever. The idea is that we're not using stats to test how good our science is. We're using stats more as we'd use a microscope to try and focus in on the patterns to see what we can see. So this is the first plot we generated as we explored the data. So you can see that you can't really tell much from here. But we made some simple changes to the code that we had developed, and so we did a facet wrap. And so you'd learn more about that in the course. And you can see, oh, well, you've plotted the data each by year. And so you can see there's slightly different patterns between years, but we still can't really get a really good idea of what's going on. So we do another plotting, and we summarize the data and then put some smoothing curves on it. And so we see, okay, so there's some patterns emerging, but why aren't all the peaks lining up? The fireflies are not merging at the same time every year. So we've got week on the x-axis and captures on the y-axis. And so we wanted to find another way to plot it. And so we know from insect ecology that insects are responsive to temperature as well as time passing. And so a way to integrate the conditions that an insect is exposed to over its lifetime is degree day modeling. And so my students went and they found the Allen double sign algorithm, which is a classic way of calculating degree day accumulation. And they coded it and processed weather data from the site. So here is our model, or part of our model that allowed us to calculate degree day accumulation. And so then they replotted the data. So here's the data by week that we saw before. And then here is the data by degree day accumulation that we see now. And so we can see that the peaks are much more closely lined up with degree day accumulation than they were for week. And so that suggests to us, just through data exploration, that firefly activity is more heavily cued on temperature cues versus on photoperiod or time cues. So then we did some additional steps, and I won't go into these in detail, but we modeled the phenology of the fireflies. Then we extracted the activity peaks that we found using good old-fashioned algebra, and then looked for trends in the activity peaks, because you can see, let's see, back here, these are still not completely coordinated, like they still aren't completely synchronized, so there's probably some other factor there. And then we look for the trends, and then we explained the variation using GLM. And then we also did a sensitivity analysis, so we tested all of our assumptions by changing our thresholds to see how that affected our conclusions. And so here's our results from the model. And so from this, the students learned that you can model the mean of the activity pretty well, but you can't necessarily capture all of the variation, and this is because of the way that insects are trapped. Sort of through random movement, you're going to have 0 on one trap and 5 on the next trap, and you're never going to be able to explain that level of variation, but you can capture the mean very well. And so this is the variation in peak emergence by degree days. And so you can see there's still a fair bit of variation that we haven't accounted for. And so we decided to look at that a variety of ways through a whole bunch of different environmental conditions. And what we found was this. So the degree day accumulation at peak emergence had this polynomial relationship with precipitation accumulation. So in extremely low precipitation years and extremely high precipitation years, we actually had a delay in emergence. So this is actually a pretty nice example of the decoupling of multiple drivers of phenology on the activity of a species. And so we also found some other cool things. So we found that fireflies were more likely to be found in the alfalfa fields and the no-till field crop fields. So this suggests that they prefer habitats with only minimal soil disturbance. We also uncovered this, and we aren't really sure how to explain this. It looks like there's a 6 or so year cycle in the abundance of fireflies, and we're going to need more data to do an investigation into that. So we managed to get some pretty good output associated with this project. So my students wrote a preprint that they submitted to bioRxiv, and we wrote up a nice paper about it, which was 2 days after being published on bioRxiv. I got contacted by a reporter from Science Magazine who wanted to write a little feature on this. So that was pretty exciting. It actually made it into the print edition of Science Magazine, and it still hasn't even finished peer review. So we also submitted it to Royal Society Open Science, and so it's currently in review there. I also produced an instructor guide that allows people to pick up this course and essentially insert your own data, insert your own students, and we can work through this together. And this will show you how to instruct a course that produces a very similar result. It's still under construction. There's a few little details I'm working on. But this instructor guide is largely ready to go, so you can see the link down there. So what I learned from the process was there was an incredible amount of appreciation for data exploration and analysis, more than you'd ever expect. I had not, in fact, anticipated the interest in multivariate statistics, and so that was not in my original syllabus. But several students asked questions that really needed to be answered using multivariate statistics approaches. And so we ended up adding on some modules, and the students all came to extra classes so that we could actually do these additional activities. Another thing that has been pretty exciting is I've received the gift of data from a variety of sources. So people hearing about this course have found their sort of file drawer data sets and said, do mine next. And so we've got a variety of data sources ready for our next offering. So thank you so much for taking the time to listen to this presentation. I'd like to thank all of the people who helped contribute to this course and make it possible, especially my students, and also to give credit to the people who produced the beautiful photos that I used in this presentation. So links to all of their Flickr profiles are available right there. Thank you.